So, good morning everyone. We're here to discuss about cytokines. Next, please. So, the learning objectives for this discussion are as follows. Number one, discuss the different types of cytokines and their respective functions. And number two, identify the different types and functions of cytokines. Next slide, please. So the contents of this discussion are as follows. First is the definition of cytokines, followed by the interleukins, and then the interleukin 1 up to interleukin 33, and then the definition of interferons, and lastly, for the definition of TNFs. Next slide, please. So what are cytokines? Cytokines are small secreted proteins released by cells that have a specific effect on interactions and communications between cells. Cytokine is the general name for it, but its other names are lymphokines that pertains to the cytokines made by the lymphocytes. Other name includes monokines that pertains to the cytokines being made by the monocytes, chemokines, those that are made by the chemotactic activities, and interleukins that are made by one specific leukocyte but also affects other leukocytes. In addition, there are three actions by the cytokines. These are autocrine action, paracrine action, and endocrine action. And the next presenter will be Miss Faith Aranguren.
Thank you, Kate. So for the interleukins, to define the interleukins, they are mediators that act between leukocytes, and they are secreted proteins that bind to their specific receptors and play a role in intercellular communication among leukocytes. Like they are assigned to each family based on sequence homology and receptor chain similarities or functional properties. The next slide, we will be discussing different specific interleukins. For each type of interleukins, I will be mentioning the cell sources or the origin and the cell targets along with the major functions that are showed in the PowerPoint. First, for the interleukin 1 alpha and the interleukin 1 beta, we have the major function of induction of pro-inflammatory proteins, hematopoiesis, and differentiation of type 17 T helper cells. They are originated from the macrophages, monocytes, and dendritic cells. They are targeting T cells, fibroblasts, epithelial, and endothelial cells. Next, for the IL-1 R antagonist, they have the major function of antagonism of interleukin 1 superfamily, which is like inhibition of the functions of the IL-1 superfamily. Their cell sources are the same from the previous one, which are monocytes and macrophages, and cell targets, which are T cells, fibroblasts, epithelial, and endothelial cells. For the interleukin 2, they have the major function of proliferation of effector T and B cells, development of regulatory T cells, and proliferation and cytokine production of innate lymphoid cells. They are targeting CD4 and CD8 T cells, NK and B cells, and innate lymphoid cells. They are originated from the helper T cells. Next would be the interleukin 3, which has the functions of hematopoietic growth factor, activation of basophils and eosinophils, and differentiation of dendritic cells and Langerhans cells. They are originated from the activated T cells, and cell targets the erythroid progenitors, granulocytes, macrophages, progenitors, and others like basophils and eosinophils. Next, we have the IL-4, which originates from the T cells and mast cells and targets the T and B cells. And they have the functions of induction of type 2 helper T cells, differentiation, survival factor of B and T cells, and have the role in tissue adhesion and inflammation. Next would be the interleukin 5, which has the function of differentiation and function of myeloid cells, increment of eosinophils, and chemotactic activity and adhesion capacity, remodeling, and wound healing. They are also originated from the helper T cells, specifically the type 2 helper T cells, and they are targeting eosinophils, basophils, and mast cells. Next, please. Next would be the interleukin 6, which has the T cell differentiation, activation, and survival, and also has the B cell differentiation and production of IgG, IgM, and IgA. They are originated from macrophages, T cells, and osteoblasts. They are the target cells, specifically hepatocytes, leukocytes, T cells, B cells, and hematopoietic cells. Next would be the interleukin 7, which are originated from the stromal cells of red bone marrow and thymus. They are targeting developing B and T lymphocytes, mature T cells, NK cells, and innate lymphoid cells. They have the specific functions such as megakaryocyte maturation and naive T cell survival, along with proliferation of thymocytes. Next would be the interleukin 8 that has the chemoattractant for neutrophils, NK cells, T cells, basophils, and eosinophils. They have the mobilization of hematopoietic stem cells and angiogenesis. They are originated from macrophages and certain types of epithelial cells, such as the endothelium. They target cells such as neutrophils and NK cells. The IL-9, or the interleukin 9, has the function of T and mast cell growth factor, inhibition of type 1 helper T cell cytokines, and proliferation of CD8 T cells and mast cells. They are originated from the T cells and have targeting B, T, and mast cells. Next would be the interleukin 10, which has an immunosuppressive effect through APCs or direct effects on T cell subsets, and suppression of IgE and induction of IgG by B cells in human subjects. They are originated from monocytes and type 2 T helper T cells, and B cells. Next would be the interleukin 11, which has the growth factor for myeloid, erythroid, megakaryocyte progenitors, and plasmacytoma cells. They have the protection of epithelial cells and connective tissue. They are originated in bone marrow stroma, along with targeting cells of myeloid, erythroid, and megakaryocyte progenitors. Next, please. Next is the interleukin 12, which has the following functions. We have the development and maintenance of type 1 helper T cells, and activation of NK cells, support of dendritic maturation, and induction of cytotoxicity. They are originated from the bone marrow stroma with with the B cells and macrophages, T cells, and they target T cells and NK cells. Next would be the IL-13, which are originated also in T cells. They target B cells, mast cells, and epithelial cells. They have the functions of activation of eosinophils and mast cells, recruitment and survival of eosinophils, defense against parasite infections. Next would be the interleukin 14, which has a specific function of activated B cells. They are mostly originated from T cells and malignant B cells. They are targeting B cells other than certain leukemia cells. Next would be the IL-15, which has the T cell activation, proliferation, and activation of NK cells, prevention of neutrophils, eosinophils, and from apoptosis. They are originated from T cells and targeting cells, specifically NK cells, monocytes, and macrophages. Next would be the interleukin 16, which has the specific function of modulation of T cell response. They are originated in monocytes, lymphocytes, and specifically B lymphocytes. They are targeting cells, specifically T cells, monocytes, macrophages, dendritic cells, eosinophils, and mast cells. For the rest of the interleukins, we have Jaynee Agoncillo. Thank you.
Thank you, Faith. For the interleukin 17 is originated from cluster of differentiation for lymphocyte. Their target cell are epithelial or endothelial cells, fibroblasts, osteoblasts, monocyte, macrophages, B and T lymphocytes, myelomonocytic cells, and marrostromal cells. Their major function is they induces granulopoiesis through granulocyte colony stimulating factor, and it mimics many pro-inflammatory action of tumor necrosis factor alpha and tumor necrosis beta. Next, please. For interleukins 18, they're originated from macrophages, and their target cells are T cells, natural killer cells, macrophages, epithelial cells, and chondrocytes. And their major function is enhancement of natural killer cell cytotoxicity, and it promotes T helper 1 or T helper 2 cell responses depending on cytokine milieu. Next is interleukin 19. They are from monocytes. Their target cells is keratinocytes, and their major function are induction of T helper 2 cytokines, and they enhance production of interleukin 6, tumor necrosis factor alpha, and interleukin 10 in monocytes. For the interleukin 20, they're originated from activated keratinocytes and monocytes. Their target cells are keratinocytes, monocytes, epithelial cells, and stromal cells in skin, lung, pancreas, and breast tissues. And their function is role in skin biology. For the interleukin 21, they're originated from various lymphocytes, and their target cells are CD4 T cell, CD8 T cells, B cells, and dendritic cells. For their major function, B cell proliferation and differentiation and survival, and they help in T cell growth factor. For the interleukin 22, they are originated from activated T cells, predominantly T helper 17 and T helper 22 cells. Their target cells are keratinocytes, epithelial cells of kidney, small intestine, liver, colon, lung, and particularly pancreas and skin. For their major function is pathogen defense, wound healing, and tissue reorganization. For the interleukin 23, they are originated from phagocytic cells and macrophages. Their target cells are T cells, NK and NKT cells, eosinophil, monocyte, macrophages, dendritic cells, and epithelial cells. And their major function are enhancement of T cell proliferation and promotion of memory T cells, and activation of NK cells. Next, please. For the interleukin 24, they are originated from monocyte and macrophages, also in Th2 cells. And their target cells are cancer cells, and their major function is tumor suppression. For the interleukin 25, they are originated from T helper 2 cells and mast cells. Their target cells are T helper 2 memory cells, fibroblasts, basophils, and KT cells, macrophages, and innate lymphoid cells too. And their major function is induction of IgE, IgG, interleukin 4, interleukin 5, 13, and 19 production. Then next is interleukin 26. They are expressed in certain herpes virus transport T cells, but not in primary stimulated T cells. Their target cells are epithelial cells, which binds in heparin. And then their major function is activation and regulation of epithelial cells. For the interleukin 27, they are originated from macrophages and epithelial cells. And their cell target are T cells and NK cells. And their major function is inhibition of T helper 17 cells response through STAT1. For the interleukin 28, they are originated from nucleated cell types, particularly in dendritic cells in response to viral infection. Their cell target are tissue-resident cells, primary monocytes, myeloid and plasmacytoid dendritic cells, and lastly, CD4 cells. And their major function is play a role in immune defense against viruses. For the interleukin 29, their major function is to play important role in host defenses against microbes, and its gene is highly unregulated in cells infected with virus. For interleukin 30, next please. The new name of P28 is subunit of IL-27. Its major function is prevention and treatment of cytokine-induced liver injury. For the interleukin 31, they are originated from activated CD4 T cell and CD8 T cells monocytes. Then their target cells are keratinocyte, epithelial cell, dorsal root ganglia, eosinophil, mast cell, basophil, monocytes. And their major function is inhibition of proliferation and apoptosis in epithelial cells. For the interleukin 32, they are originated from monocyte macrophages. And their target cells are macrophages, dendritic cell, T cells, peripheral blood mononuclear cells, and monocytes. Their major function is induction of tumor necrosis factor alpha, interleukin 8 and interleukin 6, and apoptosis of epithelial cells. Lastly, for the interleukin 33, they are originated from helper T cells, and their target cells are basophil, mast cells, eosinophil, and macrophages. Their function are transcriptional repressor activity, induction ng T helper 2 type inflammation on mucosal tissues, enhance integrin expression in basophil and eosinophil, and lastly, inducer of ILS. Next will be discussed by Ms. Kathleen Rivera.
So for the uh, interferons is defined as the group of cytokines that is discovered in virally infected person. It is one of the body's natural defensive response to foreign compounds. It is also among the broadly active physiologic regulators because it enhances the expression of specific genes, it inhibits cell proliferation, and it augments or activates immune effector cells. So additionally, interferons have been demonstrated to act as antiviral agents, immunomodulators, and ateneoplastic agents. So interferons as antiviral agents cannot directly inhibit the viral entry into the host cells, but they can inhibit the replication of viral gene, which includes the RNA and proteins, and the assembly of viral particles, which thereby limiting the viral infections. So like interleukins, interferons also have two types. The type 1 interferons, which is classified into two, most important type, which is the Tnf alpha and Tnf beta, and the type 2 interferons, which is the Tnf gamma. So the type 1 INF mediates the early innate immune response to viral infections, meaning the production or synthesis of type 1 interferons are triggered by viral or bacterial infections. So as I mentioned earlier, there are two major types of type 1 interferons. They are structurally different but bind to the same cell surface receptor, and they also induce similar biologic responses. So for the type 1 interferons, which is the Tnf alpha, it is produced by the infected leukocytes and the epithelial cells in response to viral infection. The second type, which is the Tnf beta, it is produced by the fibroblast and epithelial cells during viral or bacterial infections. So the type 1 interferons are induced by the pro-inflammatory cytokines such as the IL-1 and the tumor necrosis factor alpha. So both subtypes of type 1 interferons can inhibit protein synthesis of virus infected cells by preventing translation and DNA replication. The type 1 interferons can also activate natural killer cells and increase the expression of MHC or major histocompatibility complex class 1 molecules. So moving on, the interferon gamma is the type 2 interferon. They are produced by the T helper type 1 cells and the natural killer cells. The INF gamma is the principal macrophage activating cytokine, and this serves as a critical function in the innate immunity and in specific cell-mediated immunity. So the INF gamma is essential for the proper functioning of macrophages because the INF gamma activated macrophages can only kill invading bacteria as other parasites by the production of reactive oxygen molecules. So additionally, this stimulates the expression of the class 2 MHC molecules in different types of cells. The expression of MHC class 2 molecules enhances the phagocytic properties of the cells. It also increases the antigen presenting capacity of antigen presenting cells. So moreover, the interferon gamma promotes the differentiation of the naive CD4 T cells to the helper T cell type 1 subset and inhibits proliferation of Th2 or T cell type 2 cells. It also acts on B cells to promote switching to certain IgG subclasses. It activates neutrophils. It stimulates cytolytic activity of NK cells. And lastly, it is antagonistic to interleukin 4. So the next topic would be discussed by Ms. Sophia San Jose.
Okay, so the last topic for this discussion is all about tumor necrosis factors. So to start with, tumor necrosis factors or TNF were first isolated from lymphocytes and macrophages and were named as such because they induce lysis in tumor cells. So they are the principal mediator of the acute inflammatory response to gram-negative bacteria and other infectious microbes. In addition, TNF is responsible for many of the systemic complications of severe infections, as this family stimulates gene transcription or induces apoptosis in a variety of cells. There are two principal physiologic functions of TNF or tumor necrosis factors. The first one is that they stimulate the recruitment of neutrophils and monocytes to sites of infection, and the second one is that they induce acute inflammation. In severe infections, tumor necrosis factors is produced in large amounts and causes clinical and pathologic abnormalities like septic shock. So next slide, please. So for the types of TNFs, the first one is the Tnf alpha, and it is the most prominent member of the TNF superfamily, which consists of at least 19 different peptides. It is secreted by activated monocytes and macrophages that can activate T cells through its ability to induce expression of class II MHC molecules, vascular adhesion molecules, and chemokines in a manner similar to interleukin 1. So the TNF alpha exists in both membrane-bound and soluble forms and causes vasodilation and increased vasopermeability. The main trigger for TNF alpha production is the presence of lipopolysaccharide, which is found in gram-negative bacteria. So there are two receptors for TNF. The first one is the TNFR1 or the TNF receptor 1, which is constitutively expressed on most tissues and binds soluble TNF alpha. It is the primary mediator of TNF alpha signal transduction in most cell types. On the other hand, TNFR2 or TNF receptor 2 is usually expressed in epithelial cells and cells of the immune system, and it is also activated by the membrane-bound form of TNF alpha. Overall, the TNF alpha activity is at least partially regulated by soluble forms of both tumor necrosis factors receptors. So that's all for our presentation. Thank you for listening.